Thanks, Jessica and Michael, and thanks everyone for coming to the symposium. For the next 10 minutes, I will be talking about imaging the locus coeruleus in Parkinson's disease with the 70 MRI. So, locus coeruleus is a principal site for the synthesis of noradrenaline. It's located in the posterior area of the pons and adjacent to the lateral floor of the fourth ventricle. The LC is about 1 millimeter wide at cross-sectional level, and it's 16 to 17 millimeters long along the rostral-caudal axis of the brainstem. Although the structure is really small, it has great impact on modulating arousal, stress response, as well as several cognitive functions such as attention and memory. In 2006, Sasaki first discovered enhancer signals of the LC can be detected using a T1-weighted turbospin echo sequence. Later, it was found that magnetization transfer-weighted images can also be used to image the LC. The signals picked up from those images possibly comes from the deposition of neuromelanin, which is the dark pigmentation in the noradrenergic cells that can chelate some neurotoxic factors such as iron or copper. This neuromelanin-sensitive sequence has been validated using histologic evidence showing the corresponding locations of the enhancer signal and LC neurons. Some recent studies also showed that the contrast-to-noise ratio in the LC is correlated with the local concentrations of both neuromelanin and the noradrenaline transporters. In Parkinson's disease, the LC has been already affected in the pre-symptomatic stage based on the Bragg staging hypothesis. The LC CNR can be used to differentiate PD at early stage. One study also showed that Parkinson's patients with REM behavior disorder had more severe signal loss in the LC comparing to the PD without RBD and healthy controls. This evidence together indicate that in vivo MR measurements of the LC can be very valuable as a biomarker for PD. However, it is difficult to accurately capture the anatomical features of the LC using 3T. Because of the relatively poor signal-to-noise, 3T sequence typically used a 2 to 3 millimeters slight thickness to allow the signal building up along the z-axis. This is problematic, especially for this rod-shaped structure where the signals are actually distributed and varied along the long axis. 70 MRI can provide a higher SNR and much higher spatial resolution for LC imaging. There is MT-weighted turbo flash sequence, which is fast and safe, has recently been developed and tested for 70 in healthy subjects. And we want to test this sequence in PD patients, and at the same time, we want to basically try to figure out what's the best way for LC segmentation in patients. So our team in Cambridge initially scanned 16 healthy controls and 10 PD patients using the MT sequence, and we acquired 112 slices with a very high isotropic resolution. The sequence takes about 7 minutes for each run and is well tolerant for both controls and patients. 2 repeats of the MT images were acquired and averaged to further improve the SNR. There are 2 segmentation methods available in literature for LC segmentation. One is manual segmentation, and the other one is a threshold-based semi-automated method. We firstly manually segment the LC by 2 blinded raters, and we tested the inter-rater reliability using the dice similarity coefficient. We found the manual segmentation is actually not reliable for PD patients. This is because, as you can see here from the picture, drawing the boundaries of the structure is difficult when the signal is diminished by the pathology. We then tested the more objective thresholding pipeline using 2 searching space and the threshold calculated from more comparable Pons baseline. The threshold was defined as a 4.5 standard deviation above the mean intensity of the reference region. Using this method, the LC can be more sensitively detected in patient group. However, when we did the group comparison of the contrast actually didn't look much different for patients. I think this is because this method is actually fundamentally biased by thresholding, which means some LC regions more affected by PD, they are less likely to survive the threshold. Therefore, the signal estimation would be inflated. Given that the signal is less detectable in PD, making those classic segmentation methods less reliable and less impartial. Here I'm presenting a human LC atlas that can be used to indicate the theoretical anatomical location of the LC. This atlas is based off 54 high-resolution MT images from healthy volunteers. We also defined a more specific searching area and more stringent threshold to be applied to both group template and individual MT images. For group template, by using this method, we can get this group level LC automatically segmented, and for individual MT images, by applying this method, we will get this LC probabilistic map. In this map, a brighter voxel means this voxel is more likely to survive the threshold across subjects. And this map can be co-registered with the patient's data as an independent LC mask and provide more unbiased signal estimation for the LC. And we will make the atlas available for everyone very soon. We also extended our study to include more PD patients. Our preliminary results showed that the group difference of LC contrast is most evident in the middle and the caudal subregions. Moreover, here I'm presenting an example of how to use this atlas to generate this LC intensity map. This method might be useful as we can use a more detailed methodology like voxel-wise analysis or like a multivariate pattern analysis, which could be more sensitive for the topographic signal difference within the LC. And please come to my poster 272 and Katarina's poster 539 today if you're more interested in the LC and you want to learn more about the MR sequence. And also for some interesting applications of the atlas, please go to Claire's poster at 738 tomorrow afternoon. And thank you very much for listening.